I'm splitting the nervous system up into two videos because in this first one I'm going to talk about how a neuron or a nerve cell works, and in the second one I will talk about larger organization of the nervous system. So these two cells that you see, these wonky weird looking cells, are called neurons, and they've got a bit of anatomy. So they have this cell body called a soma. Soma means body in Latin. And then we have this big long thing called an axon. And then these guys are called dendrites. So the dendrites receive signals, and the axon carries the signal along until its axon terminal or end. And when we get to the axon terminal, we are going to transmit that signal through a gap called a synapse. So somewhere in here is a gap. They overlap the cells a bit, so you can't really see it, but there's a synapse, and it's going to transmit the signal from the axon of the presynaptic cell, which is this upper left guy, to the dendrites of the postsynaptic cell, this one here. So how does this signal get propagated? Well, it has everything to do with charges and voltage. So our neurons actually have a potential or a voltage, which is just a difference between the charge out here and the charge in here, of about -70 millivolts. And this means that this part inside the cell is negative compared to that by a measure of -70 millijoules per coulomb of charge. And this is maintained by ion channels, so just proteins in the membrane that let ions flow through however they want, as well as by this active transporter called the sodium-potassium pump, or sodium-potassium ATPase. And I want to talk about this for a second because this one, it's very high yields in any biology class to have, you're definitely going to have questions about the sodium-potassium ATPase. And what it does is it uses up ATP, as the name suggests, to pump out of the cell 3 sodiums and pump into the cell 2 potassiums. So we can remember that because sodium is bigger, we're going to pump more of them out than potassium's in. And there's a nice little mnemonic device to remember which way each one goes. So it goes pump, K, which is potassium, in. It's a pumpkin. This is my really bad rendition of a pumpkin. That stem is way too big. But that's how to remember which way the sodium-potassium ATPase goes. And this, along with those ion leak channels, is going to make sure that our cell stays at -70 millivolts until, that is, we get a signal. And when we get a signal, things get interesting. So this scary-looking graph shows what we call an action potential, which is what happens when your neuron gets stimulated. And we start out at resting potential, so that's this red bit here. And it corresponds to this state, where all the stuff we just talked about is happening. When a signal comes in, it's going to make sodium flow into the cell. And sodium's going to flow into the cell, and if enough of it flows in, so that's happening here, then what happens is we reach a threshold, and when we reach the threshold, all these other sodium channels are going to open. They're called voltage-gated ion channels, so they open because we've reached a certain voltage. And this is going to allow even more sodium to flow into the cell, and it's going to make the inside of the cell very positive as compared to the outside. This is going to trigger voltage-gated potassium channels, and potassium is going to start to flow out of the cell to try to balance this out. And when it tries to balance this out back to resting potential, it overshoots a little bit. So there's a moment called hyperpolarization. And then we get back to our normal resting potential. So that's what happens during an action potential. To review, we get a signal, we get a little bit more positive inside the cell. When we reach a threshold, if we reach a threshold, it's an all or nothing. If we do, then we have sodium flowing in to make this big spike. If we don't, nothing happens. If sodium flows in and makes this big spike, potassium is going to flow out and try to fix this. And then we're going to overshoot a little bit, and then we're going to get back to resting potential. So take a few minutes, pause the video, review that if you need to, because this is super, super important. There's another mechanism I want to look at that's important for the propagation of action potentials, and this is called a myelin sheath. So just like electrical wires can be insulated, these neurons can be insulated, and they can be insulated with this substance called myelin that can wrap around the axon of this neuron. And all this does is that if there's myelin there, you can't have your ion channels there as well. I mean, where are the ions going to flow out to if their outflow is blocked, right? So that means that certain ion channels, like these sodium and potassium ion channels we're talking about, are only going to be able to be expressed on specific parts of the membrane. And the action potential is going to end up jumping along, so we call this saltatory conduction. If you know Spanish, you know saltar means to jump. Otherwise, Latin is where it actually comes from saltare. But anyway, this is saltatory conduction, where it goes from each of these nodes to the next one. These are called nodes of Ranvier. Now, where does all this myelin come from? It comes from two classes of cells. In the central nervous system, that is to say the brain and spinal cord, it is going to be made by cells called oligodendrocytes. In the rest of our body, it's going to be made by cells called Schwann cells, and they're weird little cells. They're like this, and they will literally wrap around the axon. So what happens when we get to the end? How do we get this message across the synapse? Well, signaling inside the cell is going to cause these vesicles filled with chemicals called neurotransmitters to release themselves and exocytose their contents into the outside of the cell, into this synapse. And when that happens, these neurotransmitters are going to diffuse across the synapse and bind to ligand-gated ion channels on the other side. And these ligand-gated ion channels are going to let in sodium ions and potassium ions into the receiving neuron or the postsynaptic neuron, and then an action potential is going to start in that one. And neurotransmitters are actually a pretty diverse group of chemicals. So here's a table of some of the common ones. Acetylcholine is noteworthy in the parasympathetic nervous system and at neuromuscular junctions, which we'll talk about both of those more in the next video. There are also a couple different amino acids that are neurotransmitters, and some of them are a little bit weird. Most of our amino acids are alpha amino acids, which means we have our amino group and we have our acid group, and attached to this same carbon here is going to be our side chain, and this carbon is our alpha carbon, so we call these alpha amino acids because the amino and the acid are both on the alpha carbon. Except that there are some neurotransmitters that are different, like GABA, which is gamma aminobutyric acid. So in that one, instead of being on the alpha carbon, the amino group is going to be far away from the acid group on the gamma carbon. There's also some of these ones, these biogenic amines. They've got some fun little benzene groups. And then there's some more classes that are not so well known. But those are neurotransmitters, and that is how a neuron works.